This is pharmaphorum web editor Nicole Rowley, and I am on site in Berlin for ESMO 2025. And with me today are Dr Nicoletta Brega, who is VP and Global Development Lead of Clinical Precision Oncology at Bayer, and with her Christine Roth. Nice to meet you again, Christine.
Yes, good to see you.
Who is Head of Global Product Strategy and Commercialization at Bayer. Pleasure to speak with you both today. Thanks for your time.
Thank you.
So at this year's European Society for Medical Oncology Congress, Bayer has multiple data presentations from the phase 3 ARASAFE trial and pivotal ARANOTE trial evaluating darolutamide in metastatic hormone-sensitive prostate cancer, to the ALPHABET trial and global REASSURE study of radium-223 in metastatic castration-resistant prostate cancer. However, it is the late-breaking phase 1/2 data from the SOHO-01 study of sevabertinib, or BEI 2927088, in advanced HER2-mutant non-small cell lung cancer that I want to focus on in today's conversation. So firstly, you've both had long careers in the oncological space, and I want to perhaps turn to you, Nicoletta, and ask you to describe the landscape of unmet need in HER2 mutant non-small cell lung cancer.
Yeah, thank you for this question. This is a very important question to start with, because in the past decades, there have been a lot of advances in lung cancer treatment and research, but still there are subgroup of patients that have substantial challenges. And among this group of patients, there are the HER2 mutant non-small cell lung cancer patients. The HER2 mutant non-small cell lung cancer accounts for about 2 to 4% of the lung cancer patients, but still it's a very devastating disease with very poor prognosis. And for this reason, it's really important to have the opportunity to test and develop targeted treatment for this specific subgroup of patients. In particular, if we think about the patient profile who has HER2 mutant non-small cell lung cancer patients, these are not patients that are the commonly stereotype of lung cancer patients. These are most of the time women, younger age, and often never smokers. This means that what happens is that often the diagnosis of lung cancer for these patients occurred very late, when the disease is already at an advanced stage. And at that point, the first treatment available for advanced disease is doublet chemotherapy, platinum-based chemotherapy, so non-targeted agent therapy that brings some side effects and relatively restricted effectiveness. So for this reason, it's really important to develop some more targeted therapies for these kind of patients.
Absolutely. Thank you for describing the landscape of unmet need there, Nicoletta. So my next question is, what does sevabertinib potentially offer in that space that the SOHO-01 study is seeking to show?
Sevabertinib has been developed to be specifically targeting the HER2 mutation. And the SOHO-01 study has the goal to demonstrate that this targeted approach can bring meaningful clinical benefit to patients with HER2 mutation in lung cancer. The data that we are presenting at ESMO this year are very promising, and the new data, in particular the one from the group of patients who never received treatment for their systemic disease, is particularly exciting. In those group of patients in the SOHO-01 study, we have observed 71% of response rate. And we have observed even higher response rate when we looked at subgroup of patients. In the subgroup of patients with specific tyrosine kinase domain mutations, the response rate was 71%. But in the subgroup of patients with the most frequent mutation, the YVMA mutations, the response rate went up to 78% with a median duration of response of 12 months. So this data really speaks positively in the direction of potentially having a new effective treatment for starting treatment for non-small cell lung cancer patients with HER2 mutation. This is also relevant for the patients because on top of the efficacy, the safety profile that is shown in our data is manageable and tolerable. We have not observed any interstitial lung disease, which is a concern for the treating physicians. We have observed toxicity and adverse events that are typical from the class of drugs and are manageable.
I mean, it's just amazing from my perspective. If I move to you now, Christine, what can you tell us about the evolving treatment landscape here? So evolving being the key word in that question of mine there, including Baer's own oncology portfolio at large and innovation strategy in terms of the shift to a greater focus on personalisation that we have been seeing in recent years.
Sure. So I think the HER2 program is illustrative of two things. Number one, the evolving trends in this disease landscape and our approach to innovation. So let me unpack our approach to innovation perhaps first, and then I'll circle back on how HER2 really fits what we're trying to do here. So our innovation strategy is really driven by a very simple premise, and that is the rapidly increasing and evolving patient population. So by that I mean rapidly increasing. Between now and 2040, we expect the incidence of cancer to increase by more than 50%. Younger patients are going to be diagnosed, or patients are going to be diagnosed younger, I should say. And there's an alarming trend that everyone here at ESMO is really focused on, is what is driving this trend towards cancers emerging in patients in their 50s instead of 60s and 70s, and then in some cancers like colorectal cancer, even patients in their 20s and 30s. Quite an alarming trend. And when you think about patients being diagnosed early, it often means on the good side, they're going to be eligible for multiple lines of therapy, but that also means tumors will become increasingly resistant. So when we think about our mandate for what we're trying to achieve at Bayer, is we're going to need more novel targets to personalize medicine, either based on the earlier diagnosis or to address treatment resistance. We're going to need to understand the mechanisms of resistance sooner, so that ideally, if we understand them when we're trying to develop a drug, we can engineer strategies to deal with them, to either how to find them or how to deal with them when they emerge. And then all of this also means that we need to be on the hunt for kinder medicines. And, today the paradigm is, what is an acceptable level of toxicity? Well, we need to move beyond that to say, what actually can either preserve or improve a patient's quality of life? So to enable all of this, Bayer has taken a unique approach to building a new innovation ecosystem that starts with our own in-house R&D. And then several years ago, we acquired 3 platform companies, each delivering a different opportunity to either discover new targets or deploy new modalities that we haven't had in the past. And the unique thing is they are owned by Bayer, but they operate independently. And then the third thing is we have very rich and long-standing academic partnerships with institutes like the Broad Institute, which you'll hear more about in a second. And then, of course, we are always open to external innovation, but especially where external innovation meets Bayer's strengths, like the strength of our medicinal chemistry group. We are focused on more selective tumor types. So our pipeline today and our ambition in the future is to continue to build scale in gastrointestinal tumors like colorectal, genitourinary tumors like prostate cancer, lung cancer, and then anything else that meets that intersection that I said before of the opportunity to significantly improve a standard of care coupled with Bayer's unique capabilities. And then the last piece of the puzzle is the multiple modalities that we can deploy. So Bayer is well known for our approach with small molecules, but we also have protein therapeutics. We have a cell and gene therapy engine that can be deployed if we find a target where that's the best mechanism. And also, Bayer was an innovator in the radioligand therapy with our product radium-223. So we have multiple modalities that we can go after. So when you bring this all back together and how the HER2 program is illustrative of that, this is a patient population that is typically skews younger, disproportionately female, with a poor prognosis. It was developed in collaboration with a leading academic center, the Broad Institute. We are already looking to understand potential mechanisms of disease so that we can address them when they emerge. And it's a wonderful story of how finding a kinder, gentler medicine can serve these patients who are diagnosed early and concerned about treatment resistance.
Absolutely. Let's take a pause and go, wow. That's quite a broad remit there. And I like this focus on kindness of treatment, because at the end of the day, it is about the quality of life that these patients have. Yeah, state short. So going on that theme of kindness and quality of life, which has been mentioned specifically at ESMO 2025. I know we're speaking quite early on, but what do you think in this 50th anniversary year of the Congress we're going to be seeing as the key takeaways? And I'm going to blend that with a final question of where do we hope to be in the oncological space? Where does Bayer hope to be in the oncological space in 5 or 10 years from now?
I think emerging at this Congress is expansion of opportunities to deploy innovations that may have been approved in the last few years, either to new patient populations or in new combinations. I think the next thing is looking at, the radioligand space is exploding right now. So for one, I am going to be very interested in hearing attendees' reactions to the data that we're presenting in prostate cancer, and I'll come back to that in a second. But I also think this year, more than ever, we're going to hear the patient voice, either the patient voice represented through the primary investigators who were presenting their research, or a very strong presence of patient advocates here at the Congress. So I mentioned I was, eager to hear some reaction to our own data, and in the spirit of the kinder, gentler medicines that we're trying to develop, of course, we're showing data from a post hoc analysis of the ARANOTE study, which basically shows that while we deliver excellent efficacy, we do that in a way that has adverse events that are similar to placebo, and importantly, a clinically meaningful improvement in quality of life. And then when you look at our other prostate cancer asset, radium-223, there's a very interesting study called Alphabet. And for the first time, we are combining radioligand modalities. So you have the high energy precision aspects of an alpha emitter in radium-223, coupled with a beta emitter, which is lower energy, a little bit more diffuse delivery, if you will. But we're able to show that you can combine these two mechanisms, and the mechanism is well tolerated for patients, which means it warrants further study. Very excited to hear people's reaction to those two studies.
Exciting, yes. Did you want to add anything, Nicoletta?
Going back to the space of the targeted agents, the principle of targeted agent is really to target the tumor specifically and get better efficacy, but also to reduce the amount of side effects that the patient have. And this really, precision medicine, targeted agents, targeted treatments goes into the direction of a more gentle treatment for the patients. Patient could live longer, but also live a life that can give them all the joy of having a normal life. This is really what we are aiming for.
I think that's a beautiful statement to end on, but I do just want to come back to my question on the future. Where do you think we're going to see this trajectory of gentleness, kindness, lowering toxicity in 5 to 10 years within oncology?
I think, and I hope, that in 5 to 10 years, this will be expanded more to even other tumor types. I see this advancement in some tumors so far, more than in others. I really see that this is going to be applied even larger and with different modalities. The advances of research are incredible. We have seen with our partners, the research institutes we are working with, they are really proceeding at high speed in this direction. So this is what I'm expecting to see in the future.
Yep. And for me, when I think about the future, again, thinking about this patient population that is rapidly evolving, I hope that we have more tools to drive earlier diagnosis. And like I said, to detect progression, to detect the potential for disease progression before it occurs, so we can intervene with a new therapy before the disease advances. I hope we have a regulatory environment that embraces innovation in conducting clinical trials so that we can deploy newer technologies, whether it's AI, synthetic control groups, whatever, but we can generate the evidence that a regulatory body needs for approval, but to do it in a faster way and potentially with fewer patients so that we can deliver new medicines to patients faster. And then I think the last piece of the puzzle is we still have so many well-known targets that are today considered undruggable. And so therefore we can't develop a precision approach for patients. I think there are going to be breakthroughs that will allow us to finally drug the undruggable targets. And I'm happy to say that Bayer is leading the way on all three of those points.
Thank you for that, Christine, and bringing up those very important, as yet unaddressed challenges that yet remain. Thank you both for your time today. It's been a pleasure.
Thank you.